everyone, welcome to the course on computer aided drug design. Today we will talk about another important factor that is called the blood brain barrier. Okay, we want some drugs to penetrate this blood brain barrier, drugs which treats certain central nervous system diseases, drugs which treats bacterial infection in the brain and so on, whereas we do not want other drugs to penetrate this blood brain barrier and disturb. Okay. So that is a very important concept which we are going to learn. Before that, in the previous class we looked at various bioavailability rules, Lipinski's rule of 5 it is called, Weber rule, Muge rule, permeability versus polar surface area. So these rules are very important when you are looking at oral bioavailability of molecules. Okay, many softwares do the calculations and check whether these rules are satisfied. As we go along, we are going to see some more rules as well. Okay, so what is this blood brain barrier, okay, and how important this penetration is? So this is composed of high density cells, okay, restricting passage of substances from the bloodstream. So we have the blood flow here, okay, then we have the brain cerebrospinal fluid, CBF it is called. So some things pass through, nutrients maybe, oxygen maybe, okay, but we do not want many things to pass through and disturb. So this is the neuron, these are the endothelial cells, this is called astrocyte. So it allows, of course, as I said, nutrients, oxygen, water. So basically it protects the brain from foreign substances such as viruses, bacteria that may be present in the blood. Okay, it prevent large molecules to pass through. So it shields the brain from hormones and neurotransmitters, maintaining a constant environment. This is called homeostasis. Okay, homeostasis, constant environment. So it allows essential metabolites, oxygen and glucose, okay, but blocks most molecules that are bigger, 500 Daltons or more actually, like hormones, neurotransmitters, viruses, bacteria are prevented from getting through. So of course we want drugs that treats the central nervous system to pass through. So we need to design drugs so that the structural features allow it to pass through the BBB. So we have a interesting situation, we do not want normal drugs to pass through the BBB, whereas if you are designing drugs for CNS or brain, we want those drugs to pass through. Okay. So what are the primary roles of this BBB? Like I said, it separates components of the circulating blood from neurons and so maintains the chemical composition of the neuronal microenvironment. Okay. This is a stable environment that is needed for the proper function of these neuronal circuits. Okay. So it is almost like electronic circuits inside. So as you know, you need to maintain all the conditions constant. There is synaptic transmission taking place, synaptic remodeling, it gets changed because of the environment and because of the exposure to the external environment. And then angiogenesis and neurogenesis. There is neurogenesis means neurons are generated from neural stem cells and progenitor cells. So these things are happening inside the brain and so BBB has to take care of this. It protects the brain from pathogens and endows relatively immune privilege. Okay. So what is the mechanism? It forms a barrier that limits compounds to penetrate easily. Okay. This barrier is very tight because many barriers, membrane barriers may have like holes, they are not loosely, they are loosely joined. So compounds can pass through these loose joint, whereas blood brain barrier is very tight, so there are no gaps. Restrictive physicochemical characteristics that limit passive diffusion, so it does not allow passive diffusion to happen. High efflux activity, so there are a lot of PGB type efflux systems which throws whatever compounds that enter into. Okay, it has got very high set of efflux pumps inside. Lack of leaky, like I said, lack of leaky paracellular permeation and capillary valve fenestration openings. That means whereas in other membrane you may find a lot of capillary wall fenestrations, but here you will not find any capillary wall fenestrations and especially in the BBB region. Okay, so it prevents this leaky permeation. Limited pinocytosis. Okay, what is pinocytosis? Cells take in extracellular fluid and dissolve solutes. Okay, that is called pinocytosis. It is like gobbling up, eating up. So it does not have much of this pinocytosis happening. So material cannot flow through. Okay, that is like eating up or gobbling up. It is not the permeation type of thing. And there is metabolism within endothelial cells. Okay, so compounds can get metabolized, and you need uptake transporters. That means there has to be transporters which takes these material inside. So these are the mechanisms by which permeation in BBB takes place. Okay. So what is the relationship between the chemical structure and BBB penetration? The physicochemical properties affect the passive transcellular BBB permeation. So hydrogen bonds, acceptors, donors, they play a very important role. Lipophilicity, because we have cellular membrane, so molecules have to be more lipophilic for it to penetrate rather than hydrophilic. Polar surface area has to be limited. Molecular weight cannot be very large. If it is large, it will not go. Acidity or basicity, the pKa value plays a very important role. I will show you that basic molecules can penetrate through, whereas not the acidic ones. Okay. Okay, so how does it affect? What is the mechanism affecting compounds exposure? Okay, so we have passive diffusion taking place here. PGB efflux, that throws it out. Then we have protein binding. So again the compounds get neutralized, or metabolism. You could have a lot of metabolism taking place, so the compounds get degraded. So these are preventive steps. Okay, the efflux is very high, binding to protein is also very high, metabolized products, small products which becomes harmless. Okay, and generally it is you have the passive diffusion, like I mentioned in the previous case, you do not have this okay, pinocytosis taking place here. Okay, it does many things. Okay, the BBB barrier and as well as the carrier. So what are these barrier functions? 4 main: paracellular barrier. This is formed by endothelial junctions, so it restricts the free movement of water soluble compounds. Water soluble means hydrophilic compound. Then we have the transcellular barrier, because we have very low level of endocytosis, that is gobbling up, and transcytosis. So it inhibits transport of substances to the cytoplasm. We have the enzymatic barrier. There are a lot of enzymes including acetylcholinesterase, alkaline phosphatase, gamma glutamyl transpeptidase, monoamine oxidase, and other drug metabolizing enzymes. So they keep on degrading, okay, like I showed you here, you know, metabolism, that is the enzymatic barrier. Cerebral endothelium expresses a large number of efflux transporters, okay, here large number of transporters, ABC transporters, ATP binding cassette transporters, like ABCB1 and so on, P glycoprotein. So 4 different barriers are there. Paracellular, that means it formed endothelial junctions, it restricts water soluble compounds. Transcellular barrier, low level of endocytosis and transcytosis. Enzymatic barrier, there are a lot of enzymes which degrade material inside coming in. Cerebral endothelium, there are a lot of efflux transporters. So these are the various barriers. Okay. Now carrier, these are responsible for carrying material from outside into inside, like nutrients to the brain, and so removal of metabolites. Okay, these are small lipid soluble molecules and blood gases like O2 and CO2 diffuse passively through the BBB, while essential nutrients like glucose and amino acids require specific transporter proteins in order to reach the brain. So we have proteins which are very specific for transporting the nutrients, the amino acids inside, whereas gases like CO2, O2 diffuse in a very passive manner. This is the carrier. So 2 different functions of the BBB. The barriers, lot of barriers here, and the carrier, we have carriers responsible for the nutrients through it and removal of metabolites, and gases passively diffuse into this BBB. Okay. So, what are the pathological states involving BBB breakdown? So, BBB can also break down. So, if it breaks down, what happens? People may start having lot of side effects if they are given even normal drugs, even maybe inflammatory drugs or cardiovascular drugs, because it is broken down. Stroke, for example. So, the blood brain barrier is formed by endothelial cells of cerebral microvessels. It provides a dynamic interface between the peripheral circulation and the central nervous system. So, it has got a lot of tight junctions, they are called TJ, between the endothelial cells served to restrict blood-borne substances. Under ischemic stroke, it decreases these TJ integrity. That means there could be a lot of paracellular permeability, though the tight junctions get loose. So, lot of gaps are produced, and it improves the paracellular permeability. Okay. Stroke is one. Parkinson disease, dysfunction of the BBB by reduced efficacy of P glycoprotein. That means the efflux pumps do not work very efficiently. So, whatever material come inside do not get thrown out. Pain. So, inflammatory pain alters BBB tight junction protein expression. Okay. It affects the protein expressions, and it also affects the BBB permeability. HIV, again it affects the tight junction disruption. So, there could be because the tight junction has been compromised, you may have paracellular permeability. So, all these diseases can have effect on the BBB, and hence, like I said, instead of BBB preventing, drugs, compounds, neurotransmitters from the body into, they may start allowing them. Generally the tight junctions get compromised here. Okay. So, there are rules by which we can think of designing some molecules which can be BBB penetrating or not. For example, ratio between the steady state concentration of the drug in the brain and in the blood, that is log BBB, is given by this relationship. PSA is the polar surface area, log P is the hydrophilic balance. So, if you calculate this log BBB using this equation, if it is greater than 0.3, okay, then we can say they are penetrating. If they are less than -1, they are poorly distributed. That means less than -1 means it could be very hydrophilic, right. So, they do not enter the BBB, okay, whereas if it is greater than 0.3, they enter. So, polar surface area plays a role, log P plays a role. Polar surface area is higher means the molecules are hydrophilic in nature. Log P is higher means the molecules are hydrophobic in nature. So, you can see those two play important role in the BBB penetration. This term BBB is the ratio of the concentration of the drug in the brain and in the blood. This is one data, one rule which says whether the molecule is permeating through the BBB or no. Okay, other rules are number of nitrogen plus oxygen should be less than 6 to prevent a BBB. Polar surface area 60 to 70, molecular weight less than 450, log D 1 to 3, okay, log P minus N plus 0 and N plus oxygen should be greater than 0. So, these are some other parameters which also tells you whether a molecule can be BBB permeable or not BBB permeable. Okay. So, getting drugs across BBB, like I said, you need drugs if you are treating CNS, if you are treating infections in the brain, or if you are treating brain disorder. So, we need to have intact BBBs, that is a major obstacle. So, 98% of small molecule drugs and large nanotherapeutics excluded from the BBB, like molecular weight, if it is very large, of course they do not enter through the BBB. Passage of substances across the BBB, like I said, passive diffusion, fat soluble substances dissolve in the cell membrane and cross the blood brain barrier. Okay. Example, alcohol, nicotine, caffeine, if it gets dissolved in the fat, it can go. Water soluble substances such as penicillin, they do not. Okay. BBB permeation decreases 100-fold as the surface area of the drug is increased from 52 angstroms to 105 angstroms. So, if you increase double then BBB permeation decreases by 100-fold. For example, a drug with a mass of 200 Dalton may have a surface area of 52 angstrom, whereas a drug with a 450 Dalton may have area of 105 angstrom. Okay. So, it decreases. BBB permeation decreases exponentially with the addition of each pair of hydrogen bonds added to the drug. So, if you keep adding hydrogen bonds, of course, you are increasing the hydrophilicity of the molecule. So, the permeation decreases exponentially. These are taken from this reference. Okay. So, what are the drugs that cross BBB? Like I mentioned, opioid analgesic, morphine, codeine, hydrocodone, fentanyl, heroin, all these opioid analgesic, they cross the BBB. CNS stimulants, cocaine, which is found in your coffee, amphetamines and MDMA, ecstasy, these are all drugs, and then tranquilizers which bind to the GABA receptors like benzodiazepines, barbiturates, alcohol, they all cross BBB. Antidepressants which increases levels of serotonin in the brain, tricyclic antidepressants and selective serotonin reuptake inhibitors. So, they all cross BBB. Hallucinogens like LSD, psilocybin, mescaline, tetrahydrocannabinol, some of these cannabis type of drugs, they cross BBB. Okay. Dissociative anesthetics which are NDMA antagonist, ketamine, chloroform and phencyclidine, they also cross BBB. So, a lot of drugs cross this, opium, stimulants, tranquilizers, antidepressants. So, you need also drugs to treat certain disorders. So, you need some drugs which can cross the BBB. Okay, interesting, look at this structure, this is called heroin, okay, this is called morphine. If you look at morphine and heroin, okay, heroin has got extra acetyl group here as you can see with respect to. So, there is an acetylization taking place. Okay. So, it becomes slightly more hydrophobic. So, lipid solubility increases. So, its potency is twofold when compared to morphine, okay, because it has become more lipid. So, it is lipid soluble, so it can cross the BBB. So, heroin has got twofold potency than morphine. So, slightly make the drug more lipid soluble, you increase the BBB penetration. 75% of the CNS drugs are basic, okay, 90% are neutral. So, basic drugs can penetrate BBB, acidic ones will not. Okay. You have a basic drug and acidic drug. So, these drugs can penetrate easily. So, if you are designing drugs for central nervous system, then look at more basic drugs, okay, not acidic drugs, because acidic drugs will not penetrate through the BBB. So, that is one way of designing compounds for BBB penetration or preventing compounds from BBB penetrating. Okay. Ideal compounds to treat CNS infection, that means if there are any infection in the brain region or if there are infection in the cerebrospinal region, then we need to have a lot of antibacterial, right. So, molecule should be small, is moderately lipophilic. So, lipid water partition coefficient at pH 7.4 of around 1 to 10, low level of plasma protein binding, because we do not want it to be effluxed out from BBB, volume of distribution of around 1 liter per kg, not a strong ligand of PGB, you do not want it to get effluxed out, right. So, it should not be a ligand of any of these efflux pump which are located at the blood brain or blood CSF, that is cerebrospinal fluid barrier. For example, some anti-infectives like tuberculosis, isoniazid, pyrazinamide, linezolid, metronidazole, fluconazole and some fluoroquinolines achieve a good CSF to blood ratio, okay, 1. Therefore, they are very valuable drugs for the treatment of CNS related infection. So, that is the strategy one can think about. Small molecules, moderately lipophilic, it should have low level of plasma protein binding, it should have it should not be in ligand for PGB, efflux pump, the volume of distribution should be around 1 liter per kg body weight. Okay. We have looked at now P glycoprotein, sorry, we have looked at blood brain barrier as one of the important parameters to consider. Now, let us look at this P glycoprotein efflux transporter, because it plays a very important role in effluxing drugs out of the system, whether it is in the plasma region or whether it is in the blood brain region or any other part. Okay, we look, these are around 70 kilodalton transmembrane glycoproteins. So, they throw compounds out of the system. So, drugs may, if they are get caught, and they may get thrown out. So, we need to design molecules which has low PGB binding ability. What is this? These are member of ATP binding cassette family of transporters. There are about 50 of them known, and they are found in different parts of the body. As for example, if you look at multi-drug resistant diseases, you will find lot of these ATPs subfamily. Okay. There are many different subfamilies and families are there. They pump out many foreign substances out of the cells. Okay. They are present in many tissues of the body, namely BBB, small and large intestine, liver, kidney, adrenal gland, pregnant uterus. So, the goal of it is to throw out any foreign substances which may be toxic to the system. So, if the drug gets caught, it may also get thrown out. Increased intestinal expression of P glycoprotein can reduce the absorption of drugs that are substrates for P glycoprotein. Okay. So, it reduces bioavailability. So, if there are a lot of PGB present in the intestine, so they get thrown out, so they does not get absorbed into the GI, so bioavailability of the drug may be less. So, supra therapeutic plasma concentrations and drug toxicity may result because of decreased P glycoprotein expression. Okay. If the glycoprotein expressions are low, then the amount of the drug may go up, which may be leading to higher plasma concentration or even toxicity. So, people who may have large expression of PGBs may have different concentrations of the same drug in the plasma, and those who have high expressions of PGB may have lower concentrations of this particular drug in the plasma region. Okay. The removal of toxic metabolites and xenobiotics from cells into urine, bile and the intestinal lumen. In fact, they are very important for us because they throw out all the toxic metabolites and xenobiotics from the cells. Okay. So, they are doing a good job, but drugs also can get thrown out of the system because of the PGB efflux action. Okay. The transport of compounds out of the brain across the blood brain barrier. So, they also do an important job of throwing out, like I mentioned in the previous section, the PGB family are quite abundantly found in the BBB region. Okay. So, where are the sites? It is found the prostate, all sites 42%, 13% prostate, breast, digestive systems is very high, pancreas, liver, lung, bronchus, bladder and so on. So, they become chemotherapy resistant, the patients become chemotherapy resistant because of okay, the high expressions of the PGB, and hence the drug resistance happens actually. Okay. So, you can see many deaths happen because of the resistant to chemotherapy, and here PGB plays a very important role. Okay, what are the mechanisms of resistance to anti-cancer drugs? For example, anti-cancer drugs, there is a decreased in uptake in the cells. So, there is a reduced apoptosis, altered cell cycle checkpoints, increased metabolism of drugs, increased or altered targets, increased repair of damage, compartmentalization. So, all these happen, and also we have increased efflux also. So, decreased uptake, increased efflux. So, all these are reasons why anti-cancer drugs become of no therapeutic value, and PGB plays a very important role, especially in keeping the concentration of the drug to very low value inside the cells, and hence the efficacy of the drug or the drug action becomes very minimal. So, we need to understand the action of PGB, and we need to understand how or what type of molecules can become PGB substrate and hence get thrown out, and what type of molecules can escape this PGB efflux action. Thereby, if it can escape the PGB efflux action, the concentration of the drug in the plasma region will be high, and hence it may act effectively. Okay. So, we will look at more of this PGB in the coming slides also. For example, if you look at how drugs get into cells, one is the diffusion, okay, like if you take anti-cancer drugs, this is through vinblastine, doxorubicin diffusion. Transport, okay, nucleoside analogues, so there are transporters which transport. Endocytosis, okay, so it is like gobbling up, like all your immunotoxins. Large molecules always go through this, large molecules cannot go through this diffusion and transporter. Okay. So, if you have overexpressed ABC transporters, okay, you have fewer functional uptake transporters, defective endocytosis. So, you could have problems leading to resistance, okay, because you are not, the drugs do not penetrate and reach the inside of the cell. This picture is taken from this reference given. Okay, so we will continue more on this topic of PGB, efflux pump transporters, and how they play an important role in the resistance acquired by cells, and how we need to design drugs to escape this PGB efflux transporters. Okay. Thank you very much for your time.